All right, hi everybody. Welcome to our second STEM Talks of the year. As you already know, the theme for this month is stem cells, and we're really excited to be able to spend a whole hour just talking about stem cells and things related to stem cells. So for those of you that haven't been to one of our STEM Talks before, basically how it works is we have three panelists that will just talk about their career. They'll talk about what they do on a day-to-day basis, how they got into their career, their education journey, and then after they all speak, we'll have an open Q&A period. So during that time, we're going to go over all the questions you submitted beforehand, as well as any questions you put in the chat. In addition, if you feel comfortable, you can also unmute yourself at that time and ask your question live to our panelists. So for those of you, though, that are here for the certificate of participation, as long as your first name and last name is spelled properly, that will count as your attendance. There's no external link or anything. Just make sure your first and last name is spelled properly. So for those of you that don't know what this program is, just a little bit of an intro. Basically, our team hosts events throughout the year. And if you attend 7 events, you're going to get a certificate of participation from us. So that's events like this career panel, things like an interactive activity, or maybe even coming in person to UHN during a PA day or for a classroom field trip. So everybody that participates will get a certificate of participation. And if you're in this health and wellness SHISM program, you can also get a certificate for it. So if you're not signed up, you can check out our website and sign up. I'll put information in the chat about it. But again, today's topic is a career panel on stem cells. So just so I'm not talking forever, I will just pass it off to our first speaker of the day.
So thanks everybody for joining today. My name is Brittany, and I'm currently a second-year master's student in the LMP program at U of T, and I'm working at the McEwen Stem Cell Institute. I've been here for actually already 3 years. And a little bit about myself. I was from the city of Markham, and I've been working in this lab, I told you just now, for 3 years. And I like drawing and making music, writing, and shopping in my spare time. Here's just some photos of me being in the lab with everybody else. And yeah, we do a lot of activities and also poster sessions. So I was an undergrad in the LMP program at U of T, but I continued on to grad school as well in this program. And so that means I joined the Ogawa lab when I was an undergrad, and I've led several projects in the lab in the past. But now I'm sort of just settling on one project, which I will introduce to you later on as a grad student. So the goal of our lab is to repair the damaged stage biliary diseases require the whole liver transplant, but the strategy is hindered by a shortage of other organs. And so our lab's mission is to differentiate human pluripotent stem cells, or HPSCs, into the liver parenchyma, trichocytes, and also biliary epithelial cells, which we call cholangiocytes, for cellular therapy. And for me, since our lab has already established this type of protocol for generating these biliary ductal cells, and my project is to make structures that look like the actual biliary network, as you can see on the right picture. And also we're trying to fabricate a perfusible bile duct, as you can see down here. It's a fun project. And so as a grad student, we are expected to have lab progress report meetings. And for my lab, at least, we do it weekly, but on a rotation basis. It's like every week as a student, and let's say we have 3 new students, so we rotate. Like everybody presents basically monthly almost. But we also have one-on-one progress meetings with our PI, and it varies from lab to lab. Like for example, my lab does it bi-weekly, but I know there are labs who do it weekly, or also even just once in 2 months. But pretty much our data analysis, and we collaborate with team projects. Like as you can see in the picture, we are a team and we always work together. And we also do journal clubs, which are, it's like book club for scientists. We read articles and we share our opinions on it. And we also have institute-wide progress meetings. So if you belong to, like for us, because we're in the McEwen Institute, so like we share our progress towards the entire institute as well. And as for data analysis, at least for me, who's in the wet lab, but typically happens remotely. So that means it's like homework. We don't really do it during the day, but there are times when you just spend the entire day on the analysis as well. But also, you will have new students and staff coming to the lab constantly, so you do have to teach and supervise them. But for me, it's pretty fun. I like the teacher. And why I chose this career is because I really like embryology. At least in undergrad, I found that I liked it. And also, I just found that I don't really see any other cell types that change this much. It's just a very, very fascinating process to me overall. And also, the clinical potential is very huge because it's an unlimited source of cells, right? Like right now, we can differentiate stem cells into like neural cells, heart cells, lung cells, just pretty much anything. And also, just being in this field is a great opportunity for learning and teamwork. So yeah, I really like it. And I think that's it for me. And thank you for listening.
Thank you, Brittany. Graham, you're up next. Feel free to take over screen share.
Okay. I hope everybody can hear me okay. My name is Graham, and I'm an advanced practice nurse educator at Princess Margaret. I work in the malignant hematology day unit, so that's kind of the team that I lead. And really my job as an APNE, or advanced practice nurse educator, is to make sure that the nurses have the knowledge and the skill to do their jobs properly. I'm also part of the Canadian Association of Nurses in Oncology. So that's CANO, that's like our nursing association for oncology. And I'm the director of recruitment and retention for CANO, which is kind of neat. It's all volunteer in that sense, but I do get paid for the nurse education practice. So I've only been in Toronto since 2015. I guess I can turn on my video. That might help too. So I've been in Toronto since 2015. I grew up in Kitchener-Waterloo, came here for my undergrad, and have stayed here ever since. In terms of my clinical background, because I was a nurse first, I did like 4.5 years, 5 years as an allogeneic stem cell transplant nurse, which I'll go into a little bit in detail later. But you know what? I think that it's really important for professionals, for people to have creative outlets. So I like painting. I've taught myself how to do stained glass. I like to go to the gym a lot. And in terms of these pictures, that's my dog. I can't miss an opportunity to show my dog in a PowerPoint. The middle picture is actually from a stem cell transplant that we gave. It was the second in Ontario for sickle cell anemia. Somehow it's just worked out that I've given 3 out of the 4 sickle cell transplants at Princess Margaret. And then I do like to travel. Who doesn't? So that's me being a goof at the Vatican. My next slide. Okay. So my education, I did my undergrad degree at University of Guelph. It was in human kinetics. I thought, hey, this is cool. I like to go to the gym. I was kind of sporty. I was like, I went to Guelph. I was like going to play football and then join the rowing team. And so I was pretty into sports. And then I thought, okay, this might work for physio. I worked at a physio clinic and I really didn't like it. I found it a little boring. And I don't need to say anything negative about my physiotherapy colleagues, but I'm a little ADHD. I like to be moving a lot. So I landed on nursing. So I went and did my undergrad at UT. There's a condensed program, so 2 years, and then I did my master's after that, which is another 2 years. In terms of my certifications, done a bunch of courses on complex malignant hematology, bone marrow transplant, immunology, and chemotherapy and biotherapy. In terms of like the allogeneic stem cell transplant trajectory, just to give you a little bit of background, we do these stem cell transplants for people with hematological malignancies. So that means like leukemias, sometimes lymphomas and myelomas. This is typically the type of people that we go for transplant. And when we talk about stem cells, we're talking about a very specific type of stem cells. They're called hematopoietic stem cells. And these are the stem cells that live in your bone marrow that turn into all your different types of blood cells. So your red blood cells, your platelets, your white blood cells, these all come from your hematopoietic stem cells. So if somebody has a diagnosis of leukemia, they come, they get treated, and they go through their induction, consolidation, and get put into remission. And if you're at a high likelihood of relapsing or for that leukemia to come back, you might be a candidate for bone marrow transplant. And if that's the case, we go through a donor registry, we find and we try to match your genes to somebody else's. We have a lot of different sources. We can get stem cells from the peripheral blood, from the bone marrow, or from umbilical cord blood. So typically we do peripheral blood. That's kind of the common one. And then once we have everything in a row, we've got our stem cells, then patients come, they're admitted to the transplant unit. We give them chemotherapy, we give them their stem cells, and we wait for those stem cells to grow. I think my best analogy for it is you've got a bunch of bone marrow where you have stem cells living in. They don't really work well, they're full of weeds. So we give this chemotherapy, this conditioning, and we sort of light the field on fire to get rid of the weeds. We totally demolish the field, and then rather than going through and picking them one by hand, and then we lay the seeds, which are the stem cells, and the stem cells grow into a nice healthy field. The problem with that is there's a lot of side effects. The chemo has a lot of side effects. Because we're transplanting an immune system as well, sometimes if we've heard of like organs being rejected from like a liver transplant, that's because the immune system doesn't like that liver. Well, we've kind of done the opposite with bone marrow transplant, where we've transplanted those stem cells. So those immune system cells come from somebody else and they can attack the whole body. It's kind of wild. And because we've messed around with people's immune systems, they're suppressed, they're really likely, they have a high likelihood of infection. So sepsis is really big. And then there's always the small chance that the stem cell, like the leukemia, can come back. Even though bone marrow transplant is kind of sold as a cure for many people with leukemia. I hope that's not too quick. That's like a crash course on allo transplant. So in terms of projects that I do, or how about I talk about these pictures? The picture on the bottom is what a bag of stem cells looks like. I'd say that one's probably darker than average, because when we collect blood when we collect stem cells from the blood, there's a lot of other blood components in there. There's red blood cells, there's plasma, there's platelets, and we try our best to only get the stem cells. But if we get too many red blood cells, like what might have happened here, then we get a darker bag because there's some red blood cells in there. The top picture is a bone marrow aspiration. So that's where we collect. We kind of have these big needles. You can see the size of that needle that goes into the bone and pulls out the bone marrow. We don't really do too many of these anymore to collect our stem cells. We can typically collect them from the blood. We give some growth factors and get the stem cells in the blood and then collect it from the blood. But in terms of projects, because I'm an APNE, I do a lot of focus on cancer system optimization. So making sure that how do we optimize and make sure that that the care that we're providing to these patients is the best care that they can achieve. So lots of focus right now on the acuity and how much sicker these patients are becoming. I also look at chemo delivery and different products. That's another part of my job. I do a lot of liaising with companies to see what's the best product out there. do accreditation. So we have to make sure that the hospital is accredited. Right now, next week, we have something called FACT accreditation. So we're not able to give, if we fail that accreditation, which I'm sure we won't, but if we did, we wouldn't be able to give some cellular therapy products like CAR T cells. And so it's kind of important. And also doing stuff like medical directives and getting things in order for the nursing. In terms of my research, GBV stands for gender-based violence. I'm working on a few projects, like research projects on gender-based violence in healthcare, and really a big passion of mine is health equity, particularly economic determinants of health, and seeing how that impacts the care that people get at Princess Margaret, but across the healthcare spectrum. With respect to stem cell transplant research, it's kind of broad, right? Like it's hard to, we know how stem cells work. They're pretty predictable in many different ways. I think that there's a lot that's growing with other projects, like you'll hear from Ian and Brittany. But with respect to the hematopoietic stem cells, we kind of have a good idea. So I'm looking at like clot. DVT stands for deep vein thrombosis, which is just clots in the vein, and seeing how that happens when people get admitted to the ICU for hematological malignancy patients, because their blood's kind of messed up in the first place. So seeing how the clots change depending on how sick they are and depending on their diagnosis is kind of what I'm focusing on. I've also looked at chemo regimens in the past for bone marrow transplant. So that conditioning chemotherapy to sort of clear the field is also kind of interesting, because there's a lot of changes that are happening, always new medications coming out and new research that needs to be done. In terms of a week in my life, I kind of have my hands in a lot of different pots, and this is kind of the case with a lot of nursing. So I'm part of the PM, like the leadership team at Princess Margaret. So there's always these daily meetings and practice ops and looking at how my unit is functioning. There's different working groups. We look at how to bring subcutaneous immunoglobulins into our practice, and medical directives and remote monitoring. It's all kind of neat. That's kind of that health systems optimization, and it takes a lot of coordination with many different people on the team. I was just at clinical orientation all day, teaching nurses how to insert IVs, and I also helped with the clinical externs, accreditation planning. Kind of talked about that. A lot of what I do, because I'm an educator, revolves around teaching as well. So making sure that the nurses have the knowledge to do the work that they're doing, and I think that it's a really important piece that they understand. That nurses understand the practical component and the physiological component of why they're doing some of the stuff that they do. I do a lot of personal education rounds. I'm looking at the next one that's coming up on Friday is about transplants and sickle cell disease, which is kind of neat. That's a little bit different than the leukemia pathway that I sort of described. Got some research time and making sure that if I get paid, then I can help work through some of the challenges that come up on the unit. So why I chose this career? Well, I think nursing in general is really diverse. It's, you don't have to go into stem cells. You can go into anything. You can go into cardiology. You can go into nephrology. You can go and you can kind of pick the path that you want to take. So I think that's kind of the nice thing about nursing. I think there's also a component of like passion and mission. And initially, I was really passionate. Immunology was cool, right? Stem cells were cool, and they are cool, and the patients are really sick. So that's what initially got me to stem cell transplant. And I think that there should be said, like leukemia is deadly, and what we do is a cure. But surviving the cure can sometimes be a challenge, especially with all those complications that can come up after transplant. So now I think that I'm in this leadership role. It's a mix. I get to do some health systems optimization and work on these projects, and it really gives me a space to be creative and think outside the box. But at the same time, I love the patient population, and really everything that I do is about trying to improve the quality of life and the quality of care for the patients that come through Princess Margaret in particular. Okay, I think that's all I have to say. And I think I'll hand it over to Ian.
Everyone see my slides? Awesome. Great. So hi, everyone. I'm Ian. I'm a postdoctoral fellow at the McEwen Stem Cell Institute. So I recently graduated from my doctoral studies a few months ago, and I've decided to stay at the Institute just to continue on with a few projects. So a little bit of background. So I've been here working with stem cells for about 8 years now. And I'll kind of tell you about my journey, what got me here. But one thing I'd like to point out, so I'll be talking about pluripotent stem cells like Brittany, and they're quite different than the adult stem cell populations that we just heard about. So I'll give a bit of a background of who I am, what got me here, and then we can talk a little bit more about the technology and how I use it specifically in my work. So I hail from Scarborough, just north of the downtown core within Toronto. I grew up there, spent most of my life there, went to Senator O'Connor High School, if anyone is familiar. And then in undergrad, I went to a research conference, and this is kind of what started my journey into stem cells. So I was at this conference called Canadian Undergraduate Conference on Health, and long story short, I listened to this one talk and it really hit home for me. The take-home message of the talk was using pluripotent stem cells to create tissues of the cartilage to remove joint replacements. That instantly fascinated me, and I wanted to know, can we take such a technology with regenerative capabilities like this and move it to other organ systems like the heart? So that's what really brought me to Dr. Gordon Keller's lab. So I've been working in Dr. Keller's lab since the third year of my undergraduate as a summer student. So I've been here for a total of about 8 years. But beyond kind of my pursuits in the academic sector, I also enjoy and try to strike a work-life balance. So during my graduate degree, I tried to play some intramurals. I also enjoy my time, spending my time working out at the gym. And I also enjoy kind of picking up some hobbies, like to learn new languages like Spanish and musical instruments like the saxophone. So a bit on my educational journey, like I mentioned, my undergraduate degree was done at Wilfrid Laurier University in Waterloo. And here I did a health sciences degree. That kind of gave me a broad understanding of the healthcare system, along with some of the nuances of basic science, such as biochemistry. And then that kind of led me on to the PhD, which I started in 2017 and just finished. And this is an example of us away from the lab having a little bit of fun. But really, what we're trying to do here is use human pluripotent stem cells. And my focus is the heart. So with pluripotent stem cells, these are cells that can become any cell in the human body. What we saw with Brittany's work is that they can become cells of the liver and then become therapeutics that way. But my real interest is the heart. And what we want to know is, can we repair and regenerate the damaged heart? So heart damage can occur from many different stimulus and comes in a variety of different flavors. What we want, and I think the lowest hanging fruit goal, is when you have a heart attack, you have this scarred region, and the scarred region in a muscle can't do what the muscle normally does, which is provide a contractile function. So the heart beats many, many times a day to pump all that blood throughout the rest of your body. In the presence of a scar, you reduce the function of the heart and it's no longer able to meet those needs. So what we want to do is take human pluripotent stem cells, make the muscle cells of the heart, and transplant them into that scar region so that you can restore the ability of the heart to contract. So what I have in this picture here is actually an example from our group. This is the work from Michael Laflamme's laboratory within the McEwen Stem Cell Institute. And here we have a heart. We have the left ventricle, the right ventricle. Excuse me. And what we have here in blue, where my cursor is the scar region. And within that, you see pockets of red. So that red is marking the muscle of human heart cells. And this white dots are marking the DNA of specifically human cells. So what we can see with this is evidence that within a scarred region of an animal, we can replace that scar with functional human muscle. So this really sets up the premise for our work. We know we can regenerate the human heart. And now how do we optimize this to push into the clinic? So that's really where my work came in for my doctoral studies and what I'm continuing now. So this involves really two things. One is we make the cells. So we make all the different cell types of the heart. what I showed you in the previous slide was us making the muscle of the heart. But we can also make the other cell types that exist in the heart, such as the cells of the blood vessel and the cells that keep the heart together. We can use these in different combinations to try to repair the heart, to grow new blood vessels. And really, we can start to play around with what is the way that, or what strategy will push this into the clinic. So that means delivery. How do we deliver the cells to the heart? Do we need a special kind of delivery system? Do we need supporting cells, supporting factors? So now we're kind of able to answer all these questions. So we can look at our cells. What I have here on the left-hand side, these are our heart cells. And what you see in green and red are actually standing for the muscle filaments. So the actual proteins in the heart cells that allow the heart cell to do its work and contract. So this is actually one of the ways that we confirm we make heart cells prior to using them, is that we look for the proteins that we know are expressed in heart cells. So the proteins are there that we know are in heart cells, we see them in our cells, and we kind of draw this conclusion from that and other evidence that yes, we are making heart cells. And then as you can see over here, this is typical day in the surgical OR. Generally, it's 1 or 2 of us. And we have quite an elaborate setup where we do open heart surgeries on different animals, ranging from small animals to mice, all the way to large animals with the pig. So what does a week in the life or day in the life look like? So given that in our work, we work with a variety of techniques, no day I'd say is exactly the same. You have ups and downs of kind of flows of work where you'll be doing one technique for a while, other techniques for a while. But really, it's a balance between getting data, analyzing the data, and writing up your results. And then there's a mixture of techniques in the lab space, such as working with the cells, surgery, and then dry lab techniques. So working on your computer, working with computer algorithms to analyze big data, and then analyzing the data you've gotten with different softwares. Beyond that, we have various meetings, project updates. So it's quite an interactive environment. You meet with your PI, with your other lab members to share and get feedback on your work. You're constantly in a mix between getting data and analyzing the data and kind of moving the step forward. And there's really quite I think, at least with our field, with the pluripotent stem cell work, it requires a lot of different techniques to answer one question. And this really is unique in the sense that we get to play around with a lot of different technologies that traditionally you wouldn't in a PhD. So it gives us quite a diverse range of techniques to ask and characterize how do you make the cells and what do they do. So I've already touched on this next part. It's really like why I chose this career. And really, so I have a personal connection to heart disease and our ability to design and create new therapeutics to help with patients who are suffering from heart disease. So I really believe that stem cells is the future of treating heart disease. I think that with the long lists for heart transplants, with the inability of the mechanical heart to kind of, one, us to have enough, two, to really provide a quality of life for many decades, we're really limited in our interventions to help people stop dying from heart disease. And I really believe that stem cells is the way to restore function and regenerate the heart in its entirety. I'm also a very curious person. I love the research we do. I love the questions we ask. I love the answers we get, even though there's a lot of times where you'll set up questions and don't get the results right away, which is part of the process that you learn to either hate or enjoy. And really, the way I see regenerative medicine and stem cells, I think this is ushering in a new age of medicine for multiple different organ groups and what we can do in terms of treating patients and really providing new outcomes for patients suffering with disease that we've never seen before.
We're on the screen now, right? All right, so I've gotten a lot of questions already. A lot of students were just in general asking, what are stem cells? And I think you guys explained the general theme of stem cells. But maybe we can start off with the one student that has their raised hand. I think Annabelle, I see your hand is up. Do you want to ask your question live? If so, go ahead.
Yeah, awesome. Thank you so much. I wanted to know for our researchers, do you guys ever study the quote unquote paracrine effect? The idea that instead of the stem cells actually doing anything, because I heard that there seems to be a problem that you get the same output even though you have different delivery types for each cell, the stem cell? Do you guys ever wonder about that part for your
Yeah.
research?
I can provide some clarification on that because I believe what you're talking about is kind of the recent studies that have come out of the clinic looking at adult stem cell populations. So and where the paracrine comes in from that. So I'll just give a little bit of background. So people have been injecting adult stem cells for quite a long time now, and there've been several clinical trials to see the effect of adult stem cells. Before I go any further, I'd like to just clarify that this is a different population than to the one that Graham is referred to in his work, the hematopoietic stem cell. So these are usually from circulating cells, adipose tissue, and they're quite different than the cells we make. So what they've seen is that independent of transplant location, these cells do not survive. And then what you hear about is these paracrine effects. So these cells secrete proteins, signaling molecules that act on the site of injury and provide effects, but the cells itself do not persist. So that's where the paracrine effects come from. But it is a bit different than the stuff that, or the cell types that any three of us work on.
Very technical question. Thanks, Annabelle. All right, another question. Graham, do you want to add to that?
Sure. I think, and again, because these are totally different stem cells, for hematopoietic stem cells, we rely heavily on knowing what different growth factors do, and we can predict, right? We know that like a colony, like a granulocyte colony stimulating factor will help produce more granulocytes. That's where the name comes from, and same with like erythropoietin, right? We can rely on different sort of growth factors to do certain things, and we know that they do that. So we wouldn't expect to see an increase in red blood cells because we're giving GCSF or granulocyte stimulating factor, because we just know that that's not how it works. I don't know if it gives any more context.
All right, I see another raised hand. Joan, do you want to ask your question live?
Okay, my question was, do you think that stem cell research is the key to immortality?
Can you repeat that again? The what of research?
Is stem cell research the key to immortality? I can give a short answer and the other panelists jump on. I don't think so. I mean, immortality is a very complex question. Sure, the idea of regenerative medicine has many promises, and the fact that we can potentially keep our organs healthy for a long period of time, and that will certainly have a direct correlation on increasing our lifespan. Immortality is a very tough question. Whether we can keep our neural activity and all these other factors going for a very, very long time, I think is a subject of debate. But certainly with stem cells and regenerative medicine, you can keep the organs in a relatively healthy function much more than we can now.
Really meta. I don't mean to. Brittany, do you have anything to add?
When people say stem cells, you think,
I think for a problem.
Yeah, so I was just saying that when I first heard about stem cells, I also correlated it with immortality. Yeah, but I think that's more for questions you might want to ask the geneticists, because I mean, they talk a lot about telomeres and like I believe personally that the answer might lie more in, like, it's too complex. But for the work that we do in the lab or in the clinic, at least, like Graham, it's more about the idea of renewal. Like, we have these diseased cells in the body, and you want to take them out and you replace them with new functioning cells that's still within your like a normal lifespan.
And a problem that we face daily in oncology care is, in order for the body to get ready to receive these stem cells, we do damage to it. We have to give chemotherapy to make room for these stem cells. And that chemotherapy, although helps fix a problem, it creates other problems, right? If you give chemotherapy, and depending on the type of chemo to sort of make room for the stem cells that I deal with, we damage the heart, we damage the kidney, we damage the liver. So we actually, we actually increase the risk for developing other cancers, which is kind of a sad thing. But to fix a immediate problem, yeah, that's kind of the.
Okay, I think kind of connected to this, one student was asking, what are the limitations of using stem cell research, stem cells in the clinic? Maybe we can just, I don't know, go in order. Brittany, what do you think the limitations are of stem cells?
Oh yeah, it's definitely first thing that comes to mind is research-wise, the cost. It takes so much time and money to optimize these like therapies and also the processes. And it is stem cells take the culture, the differentiation process already takes much longer than other cells. Like people work with capture 2 cancer cells, like CACO2, just like a lot of other labs work with these immortal cell lines that are really quick to divide, and they can use that for functional analysis, high throughput analysis. But for us, it takes a long time to do some research. And another challenge, you put in an animal model, but how do you know that criticizing, like, let's say we develop a genetic edited mouse model and then, okay, but how do you know this is the exact way that it happened between the wet lab in vivo and to human is, I think it's another thing that we must address.
Ian, why don't you go for it?
Sure. So yeah, let's talk about two limitations right away, just of the technology entering the clinic. So the first one being the safety signals. So although you can make the cells, getting the safety signals in the preclinical studies is something that is not straightforward. An example from my work is the heart cells. So heart cells are electrically active, and the cells that we make from stem cells are quite immature in the sense that their electrical activity is quite spontaneous. So it's not well regulated. Because of that, when you transplant them into the heart, they create arrhythmias. And you don't want arrhythmias. That's very dangerous to have in a patient. So that's a big safety signal that has been a problem, preventing the heart cells from moving into preclinical studies in humans. And this is common for many, many other different stem cell transplants where getting the preclinical safety is a problem. I think another thing is, so right now, at least what we do in the McEwen Institute, a lot of what we're focusing on is taking a single cell and replacing a single cell. And I guess the dream would be is, if we want to take a little anecdote from like Star Trek, picking up organs and having a 3D printed organ. So I think we're limited in our ability and our knowledge to construct organs, how to get the cells to rearrange, and how to create 3D architecture that we see in the human body outside of the body. So I think those are two biggest limitations we face right now, but young scientists like yourselves will be able to solve them.
You can have both organs, right? They have these cell-free organs, and then they just replace, just coat the inside, replenish the inside with cells. I know Iron Rogers does that. Do you think that could be a voucher? I mean, this is a place where opinions are also really divided, like the use of decellularized organs as a sort to provide a scaffold for our stem cells too. So that means the organ itself, the structure is already laid out, but then all we have to do is just put the cells. I just wonder what your opinions on that.
I mean, I guess a word of caution. So for the broader audience, many people have been trying to create large scale organs, and what they've been doing is they've taken animal organs such as a pig, and let's say we have a pig's heart. You remove all the pig cells, all the pig's protein, then you have this kind of fibrous matrix, collagen, a bunch of other kind of fibrous proteins that are left, and you can think of this as like a little scaffold now of the organ. And what people do is they take a liquid with the cells and run it through that scaffold in the hopes that the cells will survive and stick. And while people have shown that you can get cells to survive and stick, there's no one who has published anything close to the organization of what we see in the human body, nor at a scale that's even relevant to a preclinical study.
And in terms of oncology care, because that's really what I know, like I was saying, the big limitation for us is preparing the body and that preparative regimen and the work that needs to be done and the medications that need to be given to get the body ready to accept those stem cells. So to be honest, I think that many of my stem cell transplanting colleagues would probably disagree with me, but for me, the big thing would be to sort of figure out a way to step away from that chemotherapy and look at alternative options to get the body either ready to accept these stem cells, or we could bioengineer or strengthen the immune system in different ways to attack the cancer itself or in a different way. So we're already doing that with some things like CAR T cell therapy, which is where we take a T cell, like an immune system cell from somebody, make it express a receptor that's sensitive to a cancer, and then give it into the body and where it's happy and it'll grow and it'll reproduce and it'll hang out and it'll always be on the lookout for those cancer cells. So for me personally, I think immunotherapies and immunomedicine is probably the way of the future for oncology care in the short term, just because that's where the research is happening. But I also love my stem cells too. So
All right, let's take a step back. These are very deep questions, but a simpler question that we got is, why did you even pick stem cells? Out of all of like the fields you could have chose, why stem cells? Just take it all the way back to the beginning. Graham, do you want to go first?
yeah, they're cool. I don't know any other way to say it. They're pretty cool. They go in and you hear so much, there's a lot of hype about them too. So like to say, oh yeah, I work with stem cells, it kind of feels kind of neat. You're like, oh, I'm on this cutting edge of science. So it's kind of neat in that sense. Just it's unique. It's yeah, it's cutting edge. I think that's what brought me to it. And you know what, in terms of like the patient care from a clinical perspective, it's complex as well. You know, like we aren't dealing with just your average sort of kidney stone. Where that's my example because I had one a couple weeks ago. It was pretty awful, but you know, it was some pain medication and some hydration, and I was good to go. This is cutting edge and it's complex.
Brittany, why stem cells?
I did mention in my intro that I really like arts, and I think it's just that stem cells. I think that like ever changing property of them, and also just like you get to do a lot of immunostaining in stem cells after you transplant, or just like to verify that you are making what you think you're making. I like doing that, and it's really fun, I guess. A lot of the other labs, like I know they do bioinformatics or protein analysis, or they bake proteins that make RNAs, but it's for me, I just feel like I like watching cells change. I like cells in general already, and stem cells where you get like full on just you get to sit there and watch the cells change. Yeah, I feel like that doesn't really make sense, but That's my reasoning.
And Ian.
Yeah, well, I'll echo a lot what Graham and Britt said. But I think my main driving reason, if I would give it one reason, is really I just think the technology of stem cells is the future, and that's something I really wanted to learn and gain mastery and to help push the field to where I think it can go.
There are so many chat questions. I don't know how we're gonna answer all of them, so maybe we can try like a speed round of what a
Oh,
person.
there's like three I can hit off right
Okay,
now.
Ian, go. Finish them off.
Can stem cells be used to treat neurodegenerative diseases like Alzheimer's or Parkinson's? Alzheimer's, potentially. Parkinson's, there's already clinical trials. One is happening in Toronto with Toronto Western and Blue Rock Therapeutics. So they're in phase 1. Let's give a little synopsis of that. So it was safe. Patients had a reduction of Parkinson's symptoms. So tremor-like symptoms were significantly reduced. Now they're gonna up the dose and test that out. And there's other companies now after that signal that are pushing for phase 1 as well. So you're really gonna start seeing a push of stem cells into Parkinson's. Next one, are stem cells susceptible to rejection from the body like graft versus host? Yes, they certainly are. So in the ideal situation, we take one of your somatic cells, so any cell of your body, we walk that back to a stem cell using the Nobel Prize discovery of 2007, looking at 4 genetic factors. You get an induced pluripotent stem cell. So essentially you have your own personalized stem cell, which is pretty cool. Take that, make into any cell you want, no rejection. That's a very costly way of doing it. And what will likely happen is you'll have these hypoimmune cells in which they're edited stem cells, and they lack certain proteins. So they lack the antigens that are kind of say what I'm wearing from the body, and they overexpress a protein known as CD47, which essentially it tells your body, hey, don't eat me. And it's very effective at telling the body not to eat it. So that's probably what it will be like going forward. So designing engineered cells to avoid the immune system. And then I'll do one more and then I'll let the other panelists have some fun. Is research on embryonic stem cells allowed in Canada? Yes, it is. So there is a kind of, so there's a lot of controversy that's happened over the years. If you want to go all the way back to 2004 with the Bush administration in the US, who stopped some stem cells. Canada, we have an approved list of embryonic stem cells that we're allowed to work on in the country. And it's from that list that you're able to work with these cells. If you want to take a cell from another country that hasn't been approved, you have to get approval from the Canadian government.
That
I'm
was
just trying to
pretty.
answer questions
That was
in the
pretty.
chat. I think I typed the answer to 2 questions. I'll try to find one that's what are your positions on ethical concerns regarding fetal stem cells? So ISCR has, which is like the world sort of stem cell regulatory organization. They say that you cannot make embryos, human embryos, in the lab for more than 14 days, which is when the like the brain starts to actually develop, and that's when you consider it an actual human. But it is there is a very, I would say, at least I feel like the line is a little bit blurred, and definitely there are concerns. But any fetal stem cell that you see, they're used in manuscripts. It's usually taken from it's usually discarded from or not used by fertilization clinics, I believe. And so most of the fetal stem cells that you see people are using are ethically approved. Yeah.
So maybe I'll just throw a little
it's not like you
clarification
yeah.
on fetal stem cells.
No,
Fetal
go
stem
ahead.
cells is quite a weird term, fetal stem cells. So it almost makes you try to draw a comparison to what Graham works on with the adult stem cells, right? And fetal stem cells, in all reality, there are no fetal stem cells. What we have is at the start of human development, you have the embryonic stem cells, which can become any cell in the human body, and then we're able to mimic that with adult cells, making them induced pluripotent. So fetal stem cells should be called pluripotent stem cells, and that was what you would find in early life.
All right, Graham, do you want to answer a bunch of questions too? Like,
What's the next one here? Can stem cells be used to kind of replace or repair a body's immune system to cure diseases such as sickle cell or diabetes? Um, I don't know if I have the theoretical knowledge that Ian and Brittany have, but I do know for something like a sickle cell disease, when we give a stem cell transplant to replace this or to sort of cure it, our goal is to not necessarily fix the disorder or fix the mutation that's causing the sickle cell disease. Our goal is just to replace enough hemoglobin, like proper adult hemoglobin, so that that sickling hemoglobin doesn't continue to cause problems. So when we give a stem cell transplant, it's kind of like this weird mix where we're happy with a little bit of a balance between hemoglobin A and B and hemoglobin S, which is the sickle cell hemoglobin. So yeah, it's a little bit different. For something like diabetes, I think we'd have to look at Ian Brittany and look at pluripotent stem cells as opposed to hematopoietic, because that is a tissue versus blood. If that makes sense.
I have one quick thing to add on to that. So from Graham's work, the adult stem cells actually, if you look at the work out of Ottawa Hospital, Dr. Harold Atkins, so what he's done is he's done bone marrow transplants. So working with the same population that Graham does for MS patients. And like Graham said, that preconditioning regime, give them chemo, wipe the immune system, and then give them HSCs again, seems to have eliminated their MS symptoms. And this is, I forgot how big the cohort is, but this is an example of in like at home in Canada of stem cell transplants being used to cure immune diseases.
all right, one final question because we only have 3 minutes left. The final question is, can you give every student a 1-sentence piece of advice? 1 sentence only, though, so it's nice and short. And maybe we'll just go in order of speakers. So Brittany, you have to try first.
I wanted to say do not go into stem cells, but that's a lie. Follow your heart in deciding what you want to research. Yeah.
I like that. I just watched an episode of RuPaul's Drag Race, like the All Stars, and it was like season 6. It's like the All Star RuPaul, and they just did an address to like the graduating class of drag university or whatever they were doing, and they all had these fun words of wisdom. So maybe I should just pull from what one of them said, but. I don't know. I think that, no matter what way sort of your education takes you, I don't think there's a wrong way. If I look at me as an example, I did my undergrad in something that I'm not remotely working in. And then I sort of shifted gears and I went to nursing school. You don't have to pigeonhole yourself in a certain idea of what you want to be tomorrow. You go, you do something. If you're interested in science, you do it in your undergrad or whatever, and just expose yourself to as many different things as possible. That's how Ian found out that he was interested in stem cells. He was doing his undergrad, and then he went to a conference on stem cells. You never know what's going to pop up. So I think just go out, take some risks. Don't be afraid of what's the right and wrong thing for you, and just do what makes you happy.
That was a little bit more than a 1 sentence, Graham, but we will allow it. Ian?
I guess one thing. So after spending all the time in a PhD, one lesson that I'll take home, and I think is applicable to many aspects of life, is having tough skin in terms of some things you do may not work on the first go, but try again, and eventually with enough tenacity and passion, great things will happen.
All right. So with that in mind, I think that's the end of our session. I'm going to launch a goodbye poll for everybody here. We just want to know if you learned something new. I hope you did. We covered a lot of topics. And I'm just going to quickly just talk about our next couple of events before I end off the session. All right, so those of you that are here will hopefully know who UHN STEM Pathways is, but we host a bunch of virtual and in-person programming throughout the course of the year. So feel free to check out our website. For instance, we have a bunch of upcoming PA Day workshops coming up where you can come on site to UHN and actually see what the labs are like that we've been talking about. So check out our website, send us an email if you have any questions.